You've probably had a basic blood test before, right? You get this report back, it's full of numbers, but what's really going on in there? Well, today, we're going way beyond the basics. We're going to uncover the incredible detective story that's hidden inside a single drop of your blood, a story packed with clues that modern medicine is just now learning how to read.
Yeah, that standard blood count can feel pretty routine, almost boring, but that's because it's only looking for the most obvious culprits. So what happens when a medical case gets a little more complicated? What are the really critical clues that we might be missing? Let's dive in and find out.
Okay, so to really get what we're missing, we have to start with what we're usually looking for. Think of a standard blood count like a police lineup of the 5 usual suspects. These are the main types of white blood cells that give doctors that first general overview of your health.
And here they are, the usual suspects: neutrophils, lymphocytes, monocytes, eosinophils, and basophils. Any standard 5-part differential test is counting these 5. They are the absolute frontline indicators, right? The first ones to get questioned in any investigation into what's going on with your health.
But what about the really tough cases? You know, the medical mysteries that don't have an easy, obvious answer. Well, for that, our detectives, the hematologists, they have to dig deeper. They need to start looking for much rarer, more subtle clues. And this is exactly where the 6- and 7-part investigation comes into play.
You can see right here how the investigation just gets bigger and bigger. The 5-part test, that's our baseline. Then a 6-part analysis throws in a brand new clue, something called immature granulocytes. And the 7-part adds one more on top of that, nucleated red blood cells. Each layer just gives us a much more detailed and revealing picture.
So what in the world are these immature granulocytes, or IGs for short? Okay, think of them as rookie cops being rushed to the scene of a major crime. They're these young, still developing white blood cells that your bone marrow really shouldn't be letting out into your bloodstream unless it's under some kind of serious stress. And finding these rookie cells out in the field, that's a really big deal. Their presence can be one of the earliest warning signs of a severe infection or even sepsis. It's telling doctors that the body is in full-on panic mode, sending out reinforcements before they're even fully trained. It can even point to some serious problems right at the source, the bone marrow itself.
Now let's move on to our next big clue, the one that really builds the case: nucleated red blood cells. See, healthy, mature red blood cells, they don't have a nucleus. They get rid of it. So finding these super immature versions in an adult's blood is just wrong. It's totally abnormal. It's like finding a key piece of evidence that just does not belong at the crime scene.
All right, so we've identified the clues we're looking for, right? The IGs, the NRBCs, but how do we actually find them? We're not talking about a scientist peering through a microscope anymore. Oh no, we are talking about some seriously high-tech forensic equipment, the modern hematology analyzer. Which, of course, leads to the big question. A single drop of blood has millions of cells, so how on earth can a machine find and precisely identify these super rare, microscopic clues in just a few seconds? Well, the answer is this brilliant combination of physics and biology. It's amazing. The first technique is called 3D fluorescence flow cytometry. It's pretty cool. Basically, special fluorescent dyes get added to the blood sample, and they latch onto the DNA and RNA inside each cell. Now, since the younger, more immature cells have more RNA, they actually glow brighter. So by measuring that glow, the machine can instantly figure out how old and mature a cell is.
But wait, there's more. The analyzer doesn't stop there. It also interrogates every single cell with lasers using a technique called multi-angle light scattering. It's like a full-body scan for every cell that zips through it.
So first, a laser hits the cell at a low angle. The way that light scatters gives the machine the cell's overall size. It's like its basic height measurement.
Then, another laser comes in at a high angle to look deeper. This one measures the internal complexity. You know, how much stuff is inside the cell, like the size and shape of its nucleus.
And finally, a laser from the side gives us details on the cell's texture and its contents, like the granules inside. By putting all the data from all 3 angles together, the analyzer builds this incredibly detailed 3D profile, and it can identify each cell with just stunning accuracy.
And you know, if that wasn't already futuristic enough for you, the latest systems are now bringing in AI. This lets the machine not only count and identify all the cells, but also use digital imaging to automatically flag any cells that have weird or abnormal shapes, bringing a potential problem to a human's attention instantly. So, we have the technology, we have the clues,
But what does it all mean for a patient? Let's look at how these advanced parameters are helping doctors crack real-world medical cases. This is where it all comes together.
Let's kick things off with a parameter called reticulocyte hemoglobin, or RETHE. Now, we're not just counting cells anymore, we're actually measuring the amount of hemoglobin inside the very youngest red blood cells as they're being born.
And why is that so important? Because it helps crack the case of things like iron deficiency and anemia way earlier than before. It's like getting a real-time memo from the bone marrow about the quality of red blood cells it's producing, giving doctors a heads up weeks before traditional tests would even show a problem.
Next up, let's talk about the immature platelet fraction, or IPF. Now, platelets are those tiny little cells that help our blood clot. This parameter measures the percentage of brand new, fresh off-the-line platelets in your circulation.
And this helps solve a huge puzzle. If a patient has low platelets, is it because their body is destroying them too fast? Or is it because their bone marrow just isn't making enough? IPF tells you which one it is. A high IPF means the bone marrow is working overtime, which points to destruction. A low IPF, well, that suggests a production problem. That is a crucial distinction for getting the right diagnosis, and it's solved by one simple number.
Okay, our final piece of evidence is the count of fragmented red cells, or FRCs. And these are exactly what they sound like. They are literally shattered pieces of red blood cells.
And finding these fragments in the blood is a massive red flag. They're a critical screening tool for really dangerous, life-threatening disorders where tiny blood clots are forming in small vessels. And they are literally shredding red blood cells apart as they try to squeeze through. Detecting these FRCs can be the very first step in saving a patient's life.
So look at how far we've come. We've gone from the 5 usual suspects to these hidden clues, from laser light scattering to artificial intelligence, all just to read the incredibly complex story hidden in our blood. It really makes you wonder, if one single drop can tell us this much today, what amazing medical mysteries is it going to help us solve tomorrow?